have a little discussion on the fluid we've been talking about for the past couple of lectures about blood itself. So number one, I'm not into memorizing numbers, so don't memorize the actual dimensions of a red blood cell. But if you look at it, it's not your traditional shape of the cell you guys have learned about in maybe 211 of the biology class, like a nice little round ball. It's actually collapsed in on itself because mature red blood cells have no organelles and they have no nucleus. The benefit of that is it causes their membranes to kind of collapse in, and the importance of the shape is that this sort of concave ability increases their surface area. And surface area is important for diffusion. So with improved surface area, we get improved ability for O2 and CO2 to cross the cell's membrane. Even causes phones to fall off their travel mugs. Okay, now, having all these red blood cells, what we notice is when they travel through a capillary, which you guys will learn about in a lecture in lab tonight, those who have it, is that they line up single file. Capillaries are so small that only blood cells can go one at a time through them. And that'll be very beneficial. And we'll see why that is a bit later. All right. Overall, what is blood for? A couple of different purposes that blood can have. One would be for transportation of gases and nutrients, like your glucose and glycogen and fatty acids, deliver your CO2 and O2 to various parts of the body. So it's a good transportation system. We know that it plays a role in regulating your temperature. I mean, you guys get hot, you kind of turn pink. You bring more blood to the surface to radiate heat. When you get really, really cold, your skin turns kind of a whitish, and if you get really, really, really cold for a long time, it turns black because of necrosis, because we're sending the blood to your core. So it's really involved in helping to regulate your body temperature. We'll see that plays a really big role in maintaining your blood pH and what that pH range is supposed to be a little bit. And it plays a role in your overall fluid volume or your overall level of hydration. In other words, how much interstitial fluid do you guys have? And then a quick little discussion toward the end about blood's ability to transport antibodies for your immune system. These things called T cells and B cells that we'll discuss a little bit later. All right, so what is blood made up of? When someone says we're looking at whole blood, that means all parts of the blood thrown in one big pot. And when you take whole blood and you centrifuge it, we separate whole blood into its three major constituents. Plasma, which is the largest portion of your blood. Then the next largest portion is the erythrocytes, the whole red blood cell count. Because as they spin around, gravity pulls them down toward the bottom of our test tube. And there's a little teeny thin layer that kind of separates the plasma from the red blood cells, known as the buffy coat. That contains your white blood cells and platelets. And together these two, the buffy coat and erythrocytes, are what's called the formed elements, the cellular component of your blood. And as you can see from the diagram, most of your blood is plasma. In terms of the pH, it's going to be between 7.35 and 7.45. It's fussy, it's very narrow, so we can help maintain normal enzyme function throughout your body. So we don't want to get too acidic, we don't want to get too alkaline with all of this. All right, adult volumes for males, average 5 to 6 liters for males, and for females it is 4 to 5 liters. So why do males have more blood than females? Scott?
Because we don't have our period.
Because of what?
We don't menstruate.
Nope, not at all. And you're not pregnant either. It's a good try, good guess. Okay. That blood only involves about 80 milliliters of fluid loss anyways. So why do guys on average have more blood than females do? Muscle mass, right? They're overall have more muscle tissue, therefore requires more blood to feed them more muscle mass. And I said the average. I had a female friend in college back in the 70s, and Janelle was a power lifter. She could squat 535 pounds. Janelle was a powerhouse, and she was a softball fast pitch, right? And she could just almost outdo any other guy in the gym. Okay, well, let's break down plasma. We know that it's the major component of blood, but all I want you to realize is, in terms of the plasma component, what is plasma mainly comprised of? Water, right? And then the other parts of it, protein. So really focus on the 92% water, and the rest of it I really don't care in terms of percentages. But let's do look at the proteins of our plasma. And there's three major categories of proteins that are found in our plasma. Right? And the first group are known as albumins. And they are just large proteins that are in the blood. Those of you that have already had lab, remember we talked about BCOP, the blood colloidal osmotic pressure? Albumins are the cause of that blood colloidal osmotic pressure. Those of you who haven't had lab yet, I'll remind you of that when we get into BCOP with our capillary discussion in lab this evening. The next major group is going to be globulins. And you already recognize some of these, right? Antibodies, known as immunoglobulins, like maybe TSI, right? Thyroid stimulating immunoglobulin. And transport globulins, like TBG, right? And these are two major types of globulins that are found in blood. And then we have fibrinogen, which are just fibers for making proteins, help form clots. We have fibrinogen. Then we have globulins, which you can break down into immunoglobulins and transport proteins, or transport globulins. And then we have the albumins, just your general non-specific protein floating in your blood. All right, let's look at the formed elements part. And of our formed elements, how much of it's actually how much of it's comprised of red blood cells? 99.9%. So basically almost all of your formed elements are red blood cells. Just a little teeny bit of it are white blood cells and platelets. All right, speaking of these formed elements and all the other cells that are in there besides red blood cells, right? Your white blood cells are comprised of neutrophils, eosinophils, basophils, and lymphocytes, and monocytes. So you want to know the name, but you do not need to memorize the percentages. You have to recognize that the names are representative of white blood cells. Now, just to give you a taste of different durations of lifespans. For example, your platelets might live for an average of 8 to 10 days before they die and have to be recycled. On average, neutrophils can live for about 6 hours, just these guys. And it can actually be just minutes. But on average, it's about 6 hours. We're going to skip the rest of them and come all the way down to red blood cells. And most books have a 100 to 120 day range for how long those cells will live before they're going to break apart and have to be recycled. It is a long time. Well, how do you make all these cells? Well, whether we're making red blood cells or white blood cells, the overall big gigantic process is called hemopoiesis. That's for making blood cells. Now, I can take hemopoiesis and look at some cells that are responsible for the production, and the stem cells are known as hemocytoblasts. Hemo for blood, cyto for cell, and what do you think blast refers to?
Building.
Yeah, fibroblasts, osteoblasts, chondroblasts, right? The builders of all of these things. Well, what are the two major types of hemocytoblasts? Well, we have myeloid stem cells and lymphoid stem cells. And what do myeloid stem cells become? Your red blood cells and most of your whites. The lymphoids will become lymphocytes. That's pretty easy to remember. Lymphocytes come from lymphoid stem cells. If you want to focus on just the red blood cell production, then we're looking at what's called erythropoiesis. And this is just only about red blood cell production, which I remember, right, because RBCs is red blood cells, is involving myeloid stem cells. And that's going to happen in your red bone marrow in adults. So where would you guys, remember where you'd find red bone marrow? So the ends of your long bones, right? And then flat bones, like your sternum and your iliums and such. How many of you like to chew on the ends of drumsticks? Oh, lying, okay. Right? It's good, it's full of nutrients, right? How many of you feed your dogs those bones?
Not
Okay,
chicken bones.
if you do this. So this is a drumstick. I take my thumb and I put it at the end of the soft bony tissue. So I'll do it like this. This part goes in my dog's mouth and they can just gnaw, 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 gnaw and get to my thumb. They won't get the diaphysis where it splinters and can kill them. Just let them chew on the soft, spongy bone part. Put your thumb at the base and let them chew their little heart's content out. You might get your thumb bit once in a while. Better your thumb than getting a splinter in your esophagus. All right, what's interesting is if you guys get really, 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 really stressed in terms of red blood cell production, your yellow marrow can be converted into erythropoiesis under extreme duress. All right, let's talk about making red blood. What, Scott?
I was thinking, why? What?
Why?
Why would it start doing that?
I don't know why. I'm glad that it does. I don't know the actual reason why. I don't know that the chemical signaling mechanism, I don't think we actually do know why in terms of what you're asking for. We just have noticed that people under extreme duress have red blood cell production taking place in places we normally would not expect.
What do you mean, like after severe blood loss or something like that?
Now you're talking about starvation. If you ever read the book Heart in the Sea, the sailors that are stranded on that boat probably would be doing that with what little red marrow they would have left in them. All right, don't get scared by this. It is rather intimidating, right? Let's go to the top first and see how little we actually have to memorize. So there's my hemopoiesis, I already know that, right? Those are my two major groups of stem cells. We already had that in the previous slide, right? That's those guys right there. And now I can visually see that myeloid stem cells become most of my white blood cells and my red blood cells. So there's a visual confirmation of their path. And the same thing for my lymphoid stem cells becoming my, bless you, lymphocytes. I do want to come over here though and have you guys realize that the myeloid stem cells will first become a progenitor cell. That I do want you to know, that from myeloid stem cells, we get a progenitor. And notice that little EPO symbol right there that we'll get to in a moment. So myeloid stem cells will become progenitor cells. Then we're going to go through a couple of rounds of generations. We're going to skip a couple of rounds of development. We're going to skip a couple of rounds of development. We're going to get right down to here, right? What's called a normoblast. That is where the red blood cell will kick out its nucleus and its organelles. It takes about 5 to 7 days to get to that level of development. It's pretty rapid progression of cell cycles. So we're going to have our myeloid stem cell, create a progenitor cell, multiple rounds of generations. Finally, we get to what's called a normoblast, where it ejects the nucleus. Once it's gotten rid of the nucleus, we now refer to it as a reticulocyte, which is an immature red blood cell. Reticulocytes are immature red blood cells. About 24 hours later, they become fully adult red blood cells, your erythrocytes. That's the only path we're going to follow, right? Now, keep in mind where we have these EPO symbols just for a moment, right? I've got a little EPO symbol here and the EPO symbol here. Well, let's look at what that EPO is all about. It's known as the erythropoietin stimulating hormone. You may have heard of it in terms of sports, for athletes to be able to make more red blood cells, right? It's actually coming from your kidneys and your liver. Those are the two organs that actually produce this hormone called EPO. Now, what causes your liver and your kidneys to make EPO? Your cells becoming hypoxic, meaning a low level of oxygen. They're starving for oxygen. And they scream, and that causes the liver to release this hormone called EPO. And the kidneys will release EPO. That will then lead to our increase in red blood cell production. question to you that's now up there in purple is, what would cause you guys to have low levels of oxygen in your tissues?
Move to Denver.
Yeah, for a little bit, yeah, it would, right? So when I go to Sun Valley, it's at 6,500 feet compared to sea level, right? Any of you high altitude climbers? All right, that would trigger you to become hypoxic, and so that would trigger you to release EPO, so you can make more red blood cells. That's what we call acclimatization, getting acclimated to the altitude. And it's noticeable. I mean, during my summer breaks, when I'm done torching you guys for summer 241 and submitting your grades, I get in my car and I get beeline to Sun Valley, Idaho. I'm there for about between 6,000 to maybe as high as 9,000 feet for hopefully 3.5 to 4 weeks. First week there, I notice it. I'm a little winded, oh my God, this is a little hard hike to do. When I come back to Seattle, and I teach at Seattle Central, and I do the stairs from the first floor to the fourth floor, it's a piece of cake, no problem whatsoever. That's no longer the case. It didn't take that long, but 120 days, your red blood cells start to die, right? All those extra red blood cells are beginning to disappear on me. I'm beginning to notice it, right? It's a little more difficult now to run up and down those stairs than compared to when I first got there. Here's the path that it would actually work, right? And I like diagrams and pictures. So we're looking at, all right, decreased blood oxygen, that's your hypoxia, right? Tells the kidneys, right, that we're low on O2. They're going to release EPO. EPO goes into the blood and travels to all the flat bones, right, where I have lots of red bone marrow. And they increase their erythropoiesis, make more and more and more red blood cells. And now I've got more and more red blood cells. See how few red blood cells you have here compared to how many you have over here in the picture. And that means that my lungs can get more oxygen into my blood. That's the normal way you do it. It takes time to do this. And the fact that it takes time means you're also adding more plasma to your blood. So yes, you have EPO triggering the production of more red blood cells, but you also get more plasma to go along with your more blood cell count. Now, yeah.
Sorry.
No, good.
Does the same effect happen with the liver? They're just showing the kidney?
Yeah.
Okay.
And yeah, let's leave it at that. And that's okay. But now, how many of you ever heard an issue dealing with red blood cells and athletes? What's the topic of conversation?
The blood gets too thick and they can throw
But
clots.
what's the conversation? What's the topic?
Blood doping.
Blood doping, right? So, what is blood doping?
Blood doping is when you have blood taken, spin it down, extract the red blood cells, and re-inject them.
What's the problem with that? It seems a lot faster than this.
It just makes your blood thicker, can cause clots and kill you. Other than that, it's a really good way to get more aerobic endurance for a little while.
Yeah, I mean that's the issue about blood doping is that I'm saying my day, God. Back in the 70s, Olympic athletes would go to Squaw Valley and go to Denver in high altitude. And they spend months training high altitude, and then come down to sea level to compete and get the benefit I would get being at Sun Valley. But now push comes to shove and competition and money and sponsorships. We have these things called blood doping, which one way is that athletes would have blood drawn, as Dave said, centrifuge it, keep the red blood cells, and then put them back into me with no additional plasma. That makes the blood thicker, increasing viscosity. It's harder for the heart to pump that thicker blood around. And that's why NCAA and all these other sports regulations are so up on blood doping. Not because it's unfair, because everybody can do it and be fair then. It's at your own personal health risk. It could actually kill you. Athletes every year do die from blood doping issues. There's other ways to do it, and lots of chemistry behind it, to kind of hide and mask the synthetic hormones you're taking. But if you want to do it, just do it this way. The natural way, the slow way. Because you increase plasma and red blood cell count. There's no increase in thickness, you just have more blood overall, which is okay compared to having thicker blood. All right, let's talk about red blood cell numbers. And your book talks about a microliter. What the frick is a microliter? 1/1000th of a liter. What the hell is I cannot picture 1/1000th of a liter. But I can picture a drop of blood. That I can picture. And in that drop of blood, you will have, how many blood cells will you have, you think, in a drop of blood? I mean, I'm talking about 1,000?
Billions.
Couple million.
How many did you say?
I don't know.
Did you say bill or mill?
B.
Bill, billions,
Really
that's a
little.
bit too much.
Millions?
250 billion.
300 million?
Scott, what did you say?
250 million.
You're really, really close. 260 million red blood cells in a single red drop of blood.
How
All
many?
right. I don't know, how many did I say, Haley? 260 million in a single drop of blood.
Wow.
How many drops to a milliliter? Roughly about 15 to 1 milliliter. How many milliliters to a liter? How many milliliters, chemistry people, to a liter? 1,000, okay. That's a hell of a lot of blood cells in 1 liter of blood, then times 5. Right, that's a lot of red blood cells. You lose about 3 million a minute. But thankfully you're making about 3 million a minute as well, so, you know, they're busy little buggers. Now, the term we have to separate your blood into fractions is called fractionization. In other words, centrifuging, spinning it to create fractions, the plasma, the buffy coat, and the red blood cell part. When you do that, you want to collect what's called the hematocrit or your PCV, the packed cell volume. And it's simply just looking at, well, I thought my picture would show up. Let's go back to here. Maybe I took it out. Let's go back to here for a second. So this, right? There's my whole blood, centrifuge it, and I fractionate it. And this right here, the formed elements, is my PCV, my percent of cell volume. The PCV here is 45%. In other words, the percentage of my whole blood, how much of it is actually cells. In this case, 45% of this whole volume are cells. That's what your hematocrit measures. Do it this way, faster. And I always have nightmares about collecting hematocrits because during graduate school, this is my second year, I was collecting blood samples from my white-crowned sparrows. It was 3 o'clock in the morning, and I had to centrifuge 300 capillary pipettes of blood, which was fine. I had 60 of them spinning around, stopped the engine, lifted the lid, and I went to go collect them, and I was tired, and I decapitated 70 of my little caplets. Blood went flying everywhere. I thought, oh, fuck. That's 2 whole days of analysis of gone out the window. And all the other grad students that were depending upon this centrifuging were clapping. Because they said, you've now been baptized. We have all done it, don't worry. And I had blood all over my hands, all over the wall. They thought, is that your blood? No. Oh, damn, wish it was, and not the bird's blood, you know. But that's the way that you have to be able to get this measurement of hematocrit. Now look at the values of hematocrit, right? It's a percentage of red blood cells in the whole sample. Who has more red blood cells per whole sample in this little list up here? Males. Again, why do males have more red blood cells per unit of volume? Because they have more, which requires more oxygen, right? That makes common sense. That's why they have more red blood cells. Now, we're going to look at the range of numbers that are up here. If you get into sports medicine or anything else, you kind of know those numbers. So when I used to work in sports medicine, if an athlete came back with a hematocrit of 58, we would have problems because that's above the norm. Why is your hematocrit higher? Where have you been the last 2 months? Let me see your arms. Let me see between your toes, right? And look for maybe needle marks. Maybe they're cheating. Because that's the importance of knowing the range of normal hematocrits. Because if someone's below it, they have a red blood cell production problem. If it's above it, what hinky thing are you doing that you're not supposed to be doing? So it helps to kind of figure out, you know, where you are in the normal world.
So if they do it go to altitude and hang out and train there with.
Hematocrit's the same. They just have large blood
Because they've got
volume.
more plasma.
Yeah, and we're okay with that. Let's talk about the hematocrit and why we're actually concerned about it. I want to know your oxygen carrying capacity. How well can you carry oxygen and deliver it to your tissues? Well, the thing in your blood that actually carries oxygen is called hemoglobin. And it has a quaternary structure. Does that term sound familiar to some of you? You guys remember primary, secondary structures of proteins and tertiaries? So this globulin has 4 major components. The 2 purple ones are what we call the alpha chains, and they're opposite each other. And then the 2 blue ribbons are going to be the beta chains. So adult hemoglobin is 2 alphas and 2 betas. In the middle of each globulin is a heme structure, surrounded by the ribbons of protein. In the middle of the heme is an iron, and that is what oxygen and carbon dioxide and or carbon monoxide will attach to the iron of the heme found on hemoglobin. So if each iron can carry 1 O2, how many O2s can this cartoon hold? It can hold 4, right? Okay. Now, how many red blood cells in a single drop of blood? 160 million. How many hemoglobin molecules in a single red blood cell? 1,000, 10,000? You guys are no freaking fun. 280 million in a single red blood cell. 280 million molecules of hemoglobin in a single red blood cell. Times 260 million red blood cells per drop of blood, times 15 drops per mil, times 1,000, times 5. That is a hell of a lot of oxygen carrying capacity. Right, that is a lot of O2 that you can shove around your body. All right. But we have to compare actually fetal hemoglobin to adult because it's not the same. So we know the structure of adult hemoglobin. It's 2 alphas and 2 betas. Just reiterate that by saying adults have 2 alphas and 2 betas. Fetuses and newborns have 2 alphas and 2 gammas, G-A-M-M-A. So they have 2 gammas, not 2 betas. The importance is that we'll get into respiration, that this gamma allows the fetuses to steal oxygen from mom. Literally steal it from mom. Then as soon as you're born, your genetics begin to stop producing that gamma. And we'll talk more about that when we get into respiration and that idea. But that's the difference between adult and fetal. Right? So fetal blood has a greater desire to carry oxygen. Fetal blood has a greater desire to carry oxygen than mom.
Is that just to feed the growing tissues?
Yep. Well, actually, the way we should say it is the reason I have a greater desire is because I need to get it from mom's blood, and the benefit is I could feed my tissues. But we'll pound that horse to death when we get into respiration. Didn't I take this out of your slides? Yes, I thought so. And I took this out too, if I remember right.
Yeah.
Why is it showing up in mine and not yours? Give me a second here. What's going on? What spooky thing is happening here? Why are you showing me those? See, I do try to make your lives a bit easier by taking things like, yeah, let's be nice and take rid of that. Why is that in there? Okay, so you guys have this. This is your next slide, right? Yeah, what the hell's going on here? All right, let me give you guys a 5-minute break because it's kind of warm and hot and stuffy in here. Go out and get some legal drugs and some cool air. We'll come back and talk about the recycling of our blood supply.